featherless fowl, fluorescing bunnies, and freakishly huge cows. Welcome to the world of modern science, where sharp eyes, steady hands, and precision moves take plants and animals to new genetically altered levels. But can these new inventions end world hunger, grow human organs, change behaviors, even cure diseases? Go into the lab, inside the human body, and discover the tricks behind transgenics as extreme science produces extreme genes. One of the biggest goals of genetic modification and selective breeding is to reduce world hunger by producing more food. Like many chickens, these birds have been bred to grow fat and as fast as possible. But with quick metabolisms, their temperatures run high, with relief tough to come by. Until now. Geneticist Dr. Avigdor Kahaner has a solution.
As long as they are kept in cool environment, this is not a problem at all because this heat is dissipated through the legs, through the face, and that's sufficient. However, once the progeny of these birds are reared in the tropics in hot conditions, because the gradient in temperature between the body temperature and the ambience is so small, they can hardly dissipate the heat. Since I'm a geneticist, I was looking for a genetic solution. That means instead of cooling the environment of chickens with feathers, is helping them feel cool by just removing the feathers. What we have here is that's the original mutant. This is a female.
She's amazingly smooth.
Yeah.
It's quite extraordinary.
This is the male.
So
And as you can see, no scales on the feet.
Right.
That's why the mutation is called scaleless.
They've got little claws on the ends of their wings. You can certainly imagine them as little miniature dinosaurs.
Yeah.
Unlike their female counterparts, sexually mature males are bright red. An interaction between testosterone and light gives them their vibrant color. As for size, you're looking at it. They won't get any bigger than this.
When I started to cross birds like these ones
With the ones
that we saw. Well, they try to flap. They still know how to flap.
With his featherless little chickens thriving, Dr. Kahaner takes things a step further. He creates what looks like a massive beast of a bird.
He's huge.
This is an
He's
adult
absolutely
male.
huge.
This male is identical in body size to the males that we saw over there in the other house, which have full feathers. The only thing that we changed is that we took advantage of this gene, the scaleless gene, that eliminates or avoids the development of the feathers. And by doing this, we allow these birds to grow nicely and comfortably in hot conditions.
You're looking at 6 generations of Dr. Kahaner's selective breeding program. And in the near future, they'll be heading off to a much balmier climate.
Hopefully in the supermarkets in the tropical countries, in Nigeria, in Indonesia, where it's very hot. For these farmers, it's easier and more efficient because there's no need.
You don't have to pluck them.
That's right. There's no need to pluck them.
But if you think these chickens are big, cows are being bred that weigh in at more than a ton and stand taller than 5 feet. These are Belgian Blues, some of the largest cows on the market. How they get this big boils down to one tiny little gene, myostatin, which regulates the growth of muscles. When the gene doesn't work, the result is a monstrous moor, weighing about 1,000 pounds more than a normal cow and bulging with 20% more muscle mass. To make sure the non-working myostatin is passed on for more bulky Belgian Blues, they're artificially inseminated. The semen is analyzed sperm by sperm to choose the one scientists want. and the end result is a massive, muscular, lean animal that produces huge cuts of meat with less fat and bone than ordinary breeds. But if a juicy steak isn't what you're craving, maybe fatty acids are. These are super salmon eggs, and Joe McGonigal is the mastermind behind these fast-growing fish. The secret is transgenics, the transfer of genes from one species to another. And in this case, it's a gene that causes the fish to grow at an amazing rate.
During this period of their lives, they're growing about 4 to 6 times faster than standard salmon grow. We can take this out and we can compare it with a standard non-transgenic salmon, and you can take a look and see how they compare, both for size and for condition.
These two fish are the same age, but the one on the right is the transgenic, nearly twice the size of his tank buddy.
These fish are virtually identical, except for one has a transgene. It grows faster during this first year of life.
Salmon grow best in warm water, but when winter hits and the water temperature drops, so does the activity of the salmon's growth gene. Basically, the cold stunts the fish's growth. To get them to grow strong year-round, Joe extracts a gene from a species of fish that grows in cold water and injects it into the salmon egg. Now the salmon will grow all 12 months of the year, whatever the water temperature. And do they ever grow?
As you can see here, these are beautiful fish. There's virtually nothing wrong with them in any way. They're streamlined, they're torpedo-shaped. There is virtually no deformity. They look exactly like conventional fish. This is not like a bodybuilder situation or people using steroids in order to bulk up. These fish are essentially producing what they need internally under their own genetic control.
Joe sees himself as a farmer of the future and believes that transgenics could make salmon farming less dangerous to the environment and protect the few remaining stocks of wild salmon.
You can reduce the environmental footprint of conventional salmon farming in a couple of ways. One is that these fish are more efficient at feed conversion. They gain about 30% more weight per gram of food they eat compared to conventional salmon, and as a result, they also produce about 30% less waste. That waste would otherwise end up in the water column or on the bottom of the ocean.
But what if a transgenic salmon found its way to the ocean and bred with an ordinary fish?
The concerns that the transgene is out there and there's no calling it back, you've changed the world forever. Now, in point of fact, the fish are sterile. They can't breed at all. There's no possibility of a transgene that exists in an Atlantic salmon escaping from the species. Novelists get paid for imagining those kinds of things. Fish breeders get paid for producing a fish that can provide food into the future without the problems of over-harvesting stocks that will frankly disappear if we keep hitting them.
So there's minimal danger of the sterile salmon gene spreading, but even sterile fish could upset natural ecosystems. Next, cloning cows that are also part human. Their blood could hold the promise of life-saving therapies when Extreme Genes returns. In today's scientific world, reproductive cloning, creating genetically identical copies of animals from clone cells, can help improve and control evolution. Dolly the sheep is the most famous clone, but since then, bio and genetic engineers have cloned entire herds of sheep, cows, and pigs. But why create clones, and how do you do it? To explain the intricacies, expert Earl Hwang shows biologist Olivia Judson how it's done.
Hello.
Hi. Today we're working with bovine.
So it's a cow egg.
Yes. These eggs come from a slaughterhouse, and what I'm basically doing is taking out all the DNA and creating a blank for the actual cloning process to begin.
So in order to clone, you need an empty egg.
Exactly.
Earl plunges a glass pipette into the egg and suctions out the DNA. An extremely delicate operation.
It is. You have to have good hand-eye coordination and
have to be gentle with the eggs. You don't want to kill them.
So cloning doesn't require complicated chemistry, just a microscope, a very sharp glass needle, and incredibly steady hands.
And I'll go in with the pipette.
Earl inserts the pipette. Then he collects the cells containing the DNA of the cow he wants to clone.
These are cells grown from the skin of the animal that we want to clone.
Exactly.
It amazes me that you can put a cell from a grown-up animal into an empty egg and something happens.
Well, the cell basically contains the whole genetic makeup of that animal.
Earl inserts one cell of the DNA into each of the empty eggs.
You can see there's a little pocket of space.
What I'll do is puncture the egg and just carefully deposit the cell in there.
Now the egg containing the new DNA gets a spark of electricity to wake it up. Earl makes it look easy, but cloning is actually pretty tough to pull off. Dolly the sheep was the only survivor of almost 300 attempts. Mastering the cloning process can bring huge rewards. The perfect animal can be cloned time and again, ultimately opening a world of possibilities. Almost every cow here is a clone, a copy of two original super cows, created by genetic engineers to save human lives. The geneticist behind these calf creations is Dr. James Roble. And he's armed these cows with special genes that could help cure some of the world's most dangerous diseases. They're actually part human.
So what is she? What makes her special?
Well, this is a calf that has a series of gene knockouts, and she also carries the human microchromosome that has the human antibody genes.
So she can make human antibodies in her blood?
Yes.
Humans produce antibodies to fight disease and destroy lethal infections like SARS and poisons like botulism. But mass-producing human antibodies has been difficult until now. Dr. Roble estimates that each of his cows is worth $500,000 because they have human DNA inside them, a partially human immune system. And now they're being milked for their blood in one of the world's most unique extraction parlors. For 2 to 3 hours almost every week, the cows are connected to pumps that filter their blood and extract human antibodies. If these antibodies were readily available on a large scale, lethal diseases like anthrax and Ebola might be eradicated.
If we want to make antibodies to treat a specific disease, what we'd really like to do is to inoculate you over and over again so that you develop a very high titer of antibody against that agent. But we can't do that. We can't treat people that way.
So if we wanted to make antibodies against anthrax, we can't, because we can't go around infecting people with anthrax.
That's correct, yes.
Which is why Dr. Roble uses cows to create antibodies instead of humans. More than 10 times our size, his goal is to turn the cows into high-volume human antibody production machines.
The ultimate goal is to, say, take anthrax, expose the cow to anthrax, inject it with anthrax, at which point it starts making lots of antibodies against anthrax. And then you can collect those antibodies, purify them, and treat people who have been exposed to anthrax.
But getting cows to produce human antibodies isn't easy. The cow's immune system has to be genetically modified to make it partly human. To do that, Dr. Roble copies 2 sets of genes from human DNA and then combines them into a package called a microchromosome. Then the microchromosome gets inserted into the cow. But first come the mice and chickens.
Take the microchromosome and you transfer it from mouse cells to chicken cells. Okay? And this is a specific chicken cell line that allows you to cut and paste chromosomes. And the best way to carry this chromosome is actually in a hamster cell line. So then it goes to a hamster cell line. We then transfer it to the cow cell line.
So let me just get this straight. To make a human chromosome, you have to go through mice, chickens, hamsters, and then cows.
And then finally cows, yes.
Wow.
Because each cell type has its own peculiar but specific quality that allows us to do a specific kind of manipulation.
And this means that they will make human antibodies in their blood.
That's correct.
Dr. Robles' human antibody-producing cows could be as important as the discovery of a new vaccination or antibiotics. Maybe one day they'll be a weapon in wiping out diseases that no antibiotic can cure, even certain types of cancer.
A cow like this, you could easily collect probably 10 liters. A cow has about 20 grams of antibody per liter of plasma. Then you're talking about 200 grams of antibody.
Dr. Robles' cows take blood donation to a whole new level. It's the kind of advance that only the new innovations of genetic engineering can provide. Coming up, mice who hold the latest anti-aging secret, and bunnies who have a special gift of glowing. When Extreme Genes returns. The answer to some of today's medical mysteries might lie within our floppy-eared friends. But Dr. Louis-Marie Houdebine doesn't breed your ordinary bugs. He creates transgenic bunnies that glow in the dark from a surprising source.
If you look at the word transgene, this means that a gene was transferred, a foreign gene was transferred to the animal.
So which gene have they got?
Well, it comes from a jellyfish from Pacific Ocean.
So Dr. Houdebine takes a gene from a jellyfish and puts it into a rabbit. But why? What does the gene do?
The gene gives a green color to the jellyfish and then green color to the rabbits.
So the rabbits glow green in the dark.
Yes, absolutely.
Can I see?
Oh, sure.
Charged with blue ultraviolet light, the rabbit's skin and eyes glow fluorescent green, just like jellyfish. This is transgenics in action. And the jellyfish gene at work in the rabbits is the GFP gene, which makes jellyfish glow. Here's how it works. Dr. Houdebine takes the jellyfish GFP gene and inserts it into bacteria. As the bacteria reproduce, the gene multiplies. Then the genes are injected into the fertilized eggs of a rabbit, which are ultimately planted inside the mother. As the eggs grow to form the rabbit embryos, the fluorescent gene is copied into every cell. 31 days later, you've got fluorescent bunnies. Despite his many genetically modified offspring, Dr. Houdebine still has a passion for the transgenic process.
I find it still impressive. You know, that tube, very small, and you have a very, very tiny drop.
Yes.
And this is the gene itself that was injected in these animals. And with that small quantity that you can see,
Which is absolutely tiny.
You can micro-inject hundreds of embryos.
It may seem strange to make glow-in-the-dark bunnies, but they're actually helping medical researchers across the world track the movement of cells. The jellyfish genes glow like a fluorescent marker, and by tagging certain cells, scientists can track where cells go after an organ transplant. These little green glowers just may help in treating problems like blindness and bone disease. But the relationship between genes and disease in humans is complex, so scientists turn to even smaller animals, mice, for answers.
Mouse is a good model for the human. Although it's small and furry, and we're big and we're not very furry, we know that its physiology, its biochemistry, the way it works is very similar to humans.
That's because mice and humans share more than 90% of their genes. To find out how mice genes tick, they're injected with a chemical that randomly alters the DNA in their sperm. all of these male mice have been given a course of 3 injections that damages their sperm. When their sperm regenerates, they should all be carriers of genetic mutations. It's time to mate. The mutated males are put in cages with the females. About 3 weeks later, the wriggling and plentiful offspring are checked to see if they have any strange new traits. If an unusual feature is discovered, scientists need to know the exact gene that's causing it. From cancer to dominant behavior, they perform an exhaustive search, and for good reason. Studying genes that cause disease in mice may provide new information on how genes cause disease in humans. Take alcoholism. These alcoholic mice lack a crucial piece of neurochemistry, so they tend to be jittery and irritable all the time. Some believe that drinking alcohol makes the mice feel more relaxed. Now, scientists have identified the gene that causes this reaction. And it's a finding that could prove enormous.
There's no doubt that if we understand more about the relationship between gene and disease, we're not just understanding the symptoms, we really understand the mechanism of what's causing a person to drink too much alcohol.
This kind of understanding could also lead to a new era of personalized medicine.
What we want is to understand the relationship between genes and disease so that we can intervene, not at the level of the gene, but at how the gene is affecting the biology of that particular person, and how we can intervene with drugs or even just better understanding of whether they're predisposed to a particular disease so we can give lifestyle advice.
But not all discoveries happen on purpose. A routine mouse tagging exercise turned up some regenerative rodents.
We had gotten a group of these mice. We ear punched them. 3 weeks later, when I came up to look at the mice, the ear holes had absolutely disappeared. We were beginning to be very excited, but we had to redo it. So we ear punched the mice again, and then we watched them for 3 weeks, and sure enough, the holes got smaller and smaller and disappeared.
But the holes didn't disappear the way you'd think, leaving a small scar behind as evidence. Instead, their ears regenerated as if the injury had never happened. Kind of like an amphibian that can regenerate. Cut off a salamander's leg and it'll grow back. So somehow these mice have the same ability. But why? Maybe it's because they're mutant mice, a variation of a type often used in scientific research. When they were created, somehow, some way, their regenerative abilities were switched on.
When I saw the pictures of what was happening in amphibians, I realized that this was, very similar to what was happening. And I actually spoke to people about it, and I said, well, what about mammals? They said, well, mammals don't regenerate.
But obviously, some mammals can regenerate. And if Dr. Heberkatz's mice can do it, maybe other mammals can too. The same regeneration genes in these mice may even be found in humans. These mice aren't growing back entire limbs yet, but their hearts have been put to the test, and the results are extraordinary.
Besides the ears, we've looked at the heart and made injuries to a small portion of the heart, and the heart heals, regenerates, function comes back, the architecture comes back, and they heal almost perfectly.
If the genes behind this regenerative power can be identified, there could be enormous medicinal benefits.
There are a lot of applications for this. I mean, hopefully, we will be able to regenerate tissues, organs in all mammals, in humans. The other possibility is that we will be able to replace cells all the time, so that we won't have aging populations of cells.
So humans still have a way to go before they can regenerate cells. But the growth of replacement organs outside the human body is on the rise. This is a bladder. And here, tissue engineers use millions of human flesh cells to grow an experimental nose. Growing skin cells and organs in the lab can have a medical impact in the future.
Because there's some really big challenges out there where patients need new bone or cartilage for joints to be grown. And that's really where I think this science can make the biggest impact and is making the biggest impact.
One day, tissue engineers could be healing burns and repairing broken bones better than surgeons.
It depends who you are, but I think if you're a patient who had a major break of a bone that wasn't re-healing, what would be exciting to you would be to regenerate that bone. The alternative therapies are very poor, and this really offers an opportunity for the first time for man to actually regenerate and cure those patients.
Next, these sheep could help thousands in need of organ transplants. And advances in stem cell research are giving some a new lease on life. When Extreme Genes returns. These sheep may look normal, but what you don't see on the surface lies just beneath their woolly coats. These are humanized sheep, part sheep and part human, and they've been created to grow replacement organs for human transplant. But it's no easy process, especially for the patient. Scientists extract a sheep embryo from the mother's womb, inject it with human stem cells, and then sew it back in. About 5 months later, a humanized lamb is born, containing both sheep and human cells. This project is 30 years in the making for Professor Ismail Zanjani.
So what is a humanized organ?
A humanized organ would be an organ in which at least part of the organ is derived from human cells.
And Dr. Zanjani wants each of his sheep to be precisely matched to each human donor, the patient who provides the original stem cells.
We would go in and take about a couple of ounces of bone marrow cells from that patient. We isolate the stem cells from them, transplant them into the sheep fetuses, and then within 2 months we will have a liver that's at least 10 to 15% made up of the liver cells derived from that patient.
But the process is far from risk-free. For instance, if DNA from sheep cells and human stem cells mixes together, the result could be cellular fusion, producing a strange new hybrid that has features and characteristics of both sheep and humans. It'd be the oddest farm creature around. But Dr. Zanjani defends the process.
In this model, transplanting the cells into the fetal sheep at this early stage does not result in fusion at all. We have not seen one single fused cells. So we either have sheep cells or human hepatocytes or liver cells.
So they maintain their identity as sheep?
They're regular sheep, they behave like sheep, they don't speak like humans, they don't behave like humans, and they don't feel like humans. That's very important.
Once the cells are injected into the sheep, they travel through the lymphatic and circulatory system. Then they're distributed to all the organs in the sheep's body. Ultimately, Dr. Zanjani hopes to farm thousands of humanized sheep to produce organs for transplant.
The nice part of all of this is that the 2 ounces of stem cell or bone marrow cells you get from these patients provide enough stem cells to do maybe about 10 fetuses.
Wow.
So it isn't just you have one organ for transplant purposes. You can have many of those animals are still available in case the first one you give fails.
Dr. Zanjani's creation of humanized sheep is still under development, so harvesting their organs isn't an option. One day, these woolly beasts may benefit thousands, unless someone else beats them to the punch. Scientists are using stem cells to save lives and grow organs. And here, saving lives starts with new lives.
It's a boy.
Dr. Nico Faraz is carrying a bowl full of scientific gold, the umbilical cord of a newborn baby. It's packed with stem cells, which are helping to revolutionize modern medicine.
Stem
Stem cells are really exciting because they have the possibility to build many cell types of the body. It just opens huge possibilities.
Stem cells are the building blocks of the human body. Starting inside the womb, they develop into the tissues that create our entire bodies: bones, muscles, brain, and skin. And stem cells can actually become any part of the body, which saves lives like little Ross's, a baby born with a life-threatening immune disease.
What sort of future would he have been facing?
He wouldn't have had a future, basically. He just wouldn't. And basically, SCID babies don't live past a year old.
SCID stands for Severe Combined Immune Deficiency. In basic terms, Ross can't produce T cells, which fight off even the smallest infections. So doctors transplanted specifically matched stem cells into Ross's bloodstream, where they multiplied and transformed into the life-saving T cells he needed.
He wasn't able to fight off any infections. So basically, he would have been dead by a year.
So even a common cold could have killed him?
That's correct, yeah. Or a kiss or anything.
Hello.
Stem cell therapy saved Ross's life. But the controversy remains heated. One of the main sources rich with stem cells is embryos. But they can also be extracted from sources like bone marrow and umbilical cords. And Dr. Faraz is no stranger to using stem cells from umbilical cords to further his work. He's going to grow one of the most complex parts of the human body from scratch: a liver.
So you want to make a little piece of liver tissue that can actually work as a liver?
Absolutely. In the long term, we can envisage maybe creating enough liver tissue to assist a patient with a liver disease or liver toxic crisis. The difference between life and death in a patient with liver conditions is only 5%. So 5% improvement can keep them alive for a decent amount of time.
Step 1. Get access to a machine that mimics the effects of weightlessness.
So this unassuming package is the bioreactor.
That's right.
This is the anti-gravity machine.
That's correct. Impressive, isn't it?
Designed by NASA, the bioreactor produces the effects of being in space, which allows stem cells to grow in all 3 dimensions.
A bioreactor is actually mimicking microgravity. When you create tissue in 3 dimensions, you get a lot of forces and physical stress on the cells that you are actually building in 3D. So this machine suppresses those nasty effects on the cells and enable us to grow liver tissue in 3D from cord blood stem cell.
Step 2. Load up the bioreactor with stem cells and persuade them to make a liver. The magic is in the growth factors. They convince the stem cells to start growing and tell them what to turn into.
So this is kind of stem cell medicine.
Stem cell medicine, stem cell soup to make them grow.
So if you have stem cells, some potent chemicals, and a bioreactor, you just might have a production line for human organs on your hands.
Turn it on.
Starts rotating.
After just 4 weeks, Dr. Faraz has a 3-dimensional chunk of human liver, and it works. It's only a few millimeters across, but in the future, tissue like this could grow into entire organs and save lives. Next, this guy is going green to the extreme. When Extreme Genes returns. Pigs. There are nearly 1 billion of these curly-tailed squealers in the world. And for those who have a taste for them, they provide more dietary protein than any other animal. For most of us, pigs conjure up muddy images and wretched smells. But despite appearances, they're actually relatively clean animals. And they're about to get a lot cleaner.
Animal agriculture is a major consumer of the natural environment. That is, the impacts on the atmosphere, upon water utilization, and in particular, Pig agriculture or hog agriculture produces copious amounts of manure.
Put another way, pig poo is toxic. It may be a natural product, but on an industrial scale, it's a serious environmental pollutant.
Pig manure has a very high level of phosphorus within it from normal diets through normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
Pigs can't metabolize phosphorus, which is why their manure is so toxic to the environment. It runs off into water supplies, causes algae to overgrow and kills fish. But John Phillips may have a solution.
A very simple and effective solution to the environmental problems caused by high phosphorus pig manure found right behind these doors.
Meet the EnviroPig. They look like average pigs, but they're not. They have a special gene that gives them eco-friendly abilities.
These pigs are unlike any other pigs in the world. They're different because they carry a very special gene that allows them to digest all of the phosphorus in their diet. The gene is constructed in the laboratory that we use.
John created the man-made gene for the EnviroPigs from scratch. One part comes from E. coli bacteria, which produces an enzyme that breaks down phosphorus. The other comes from a mouse, which produces a genetic switch that creates a special enzyme in saliva. These two pieces of DNA combine to form a new compound gene, and then the gene was inserted into a fertilized pig egg. Now, every time John's EnviroPigs eat and produce saliva, they also make a phosphorus-busting enzyme. Their poo is now a little safer for the environment.
The aim, ultimately, we think, is that the global environment would be best served if every pig could digest all of its organic phosphorus in its diet the way these pigs do.
John's hope is that one day, all pigs will be EnviroPigs. But like most genetic experiments, it's a hot topic of debate.
There are many people out in the community that do have concerns about transgenic changes in animals. And some of these people have gone about intentionally frightening the public about this technology. So we have an uphill battle in terms of people's perceptions. This is an effective way of dealing with a serious environmental problem. It's simple, it can be propagated very rapidly. Any other technology cannot touch this in terms of the rapidity of its application on the farm or the extent of the change that can occur as a result of the technology. Nothing comes close to this.
Except maybe a genetically modified plant that can save lives. The plants in this greenhouse are contained to limit contact between them and the environment. Keeping them from open air prevents genetic pollution or cross-pollination with natural organisms. Under calibrated lights and misting machines, a new version of a crop that feeds billions of people every day is thriving: rice. But because genetic engineering is highly regulated in some countries, food like this doesn't always reach those who need it most. Here, every plant is cataloged, recorded, and meticulously looked after. For good reason. They may be able to help cure a condition that kills 2 million children every year: vitamin A deficiency. Half of the world's population gets the majority of their nutrition from rice. But rice lacks simple chemicals called carotenoids, which give color to vegetables like carrots and peppers. Without carotenoids, our bodies can't make vitamin A.
Vitamin A deficiency suppresses the immune system, but before it does that, it makes individuals blind. Most of the children who are affected are less than 5. Most of them die within 2 years of becoming blind. People suffering from vitamin A deficiency die from diseases which healthy people can survive. So vitamin A deficiency kills 2.5 million people a year. That's equivalent to a 9/11 every 12 hours. to a tsunami every month, to 250 people dying during the course of a 1-hour television program.
So the challenge for scientists was to make rice high in vitamin A that produced carotenoids. They identified 3 genes, 2 from daffodil and 1 from bacteria, that they thought would produce carotenoids. Then, they inserted the genes into rice DNA. In 1999, after years of trial and error, success. They finally created a rice that produced beta-carotene, a member of the carotenoids. The genetically engineered rice was named golden rice, after its color.
At the time, no other genetically modified plant had more than 1 gene expressing. They put 3 in and turned on a pathway.
A bowl of golden rice can provide enough vitamin A to meet daily needs. But in many countries, genetically modified foods are still locked in greenhouses. Dr. Adrian Dubach believes the benefit far outweighs the risk.
Genes occur in all the food we eat. They occur in all organisms. Biology is a most powerful tool, and there's nothing to be frightened of in this technology. The history shows it's extremely safe. It's the most regulated technology of any technology that's ever been introduced. So how can rich people in Europe stand in the way of choice for poor people in the developing world, especially when standing in the way of that choice kills them?
As with most genetic experiments, some are wary of the exploitation of this technology and the effects of cross-pollination. But not only can biotechnology save lives, it can tackle disease on a global scale, even with one of the most lethal plants on the planet, tobacco. Scientists are using it to stop the spread of one of today's most deadly diseases.
Most people associate it with cancer, and you want to make it into a plant that is going to potentially save the lives of millions.
Yeah, well, I think most people are very familiar with the concept of getting medicines from plants. We've done it for hundreds of years. But with the advent of new technology, particularly genetic modification of plants, we can take plant species like tobacco and turn them into factories for making new medicines.
So tobacco may not deserve such a bad reputation after all. Dr. Ma is using the killer weed to help stop the spread of HIV and save millions of lives.
HIV is a huge problem, of course, around the world, but particularly in developing countries, where there are millions of people who need this kind of protection, but who essentially can't afford to pay for expensive modern medicine.
So explain to me how it actually works.
Well, we're looking at production of something called an HIV microbicide. So this is a compound which inactivates the virus when it comes into contact with it.
It's called cyanovirin, and it prevents transmission of HIV. Dr. Ma wants to turn it into a cream that can be used by both men and women as a barrier against HIV infection. But the only thing that produces cyanovirin naturally is a tiny blue-green algae. And that's not much good if you're trying to mass-produce a cream for millions. So Dr. Ma has taken the gene that produces cyanovirin from the algae and put it into his tobacco plants.
So these are the plants?
Yeah, so once we've established the little plant that's downstairs, they come up and we pop them into soil. And now it's all about letting them grow, generate biomass, and also to let them go to flower and eventually go to seed, because that's where the scalability comes in. One plant will probably produce about, I don't know, 10,000 seeds.
Once Dr. Ma has created one plant that makes cyanovirin, he hopes to take its thousands of seeds and ship them around the world. Local farmers could cultivate acres of these plants, all bursting with the compound. Dr. Ma's HIV barrier cream could one day be mass-produced around the world.
Plants are cheap to grow. Once you get to this stage, you're actually in very low-tech technology. I mean, anybody can build a greenhouse very cheaply, even a contained greenhouse, and grow a number of plants. And if you really want to address global access to modern medicines, you have to think about new technologies, and that's where I think plants come in.
A single leaf on a single plant. in one corner of the farm, and the work of scientists like Dr. Ma could save the lives of millions. Genetic engineering isn't science fiction, it's reality. But unlocking the secrets behind it poses a polarizing question. Should we or shouldn't we? The debate is bound to rage on. Yet no matter what the next discovery, extreme genes